One explanation for why gene F might be expressed at high levels in some patients that would occur prior to mRNA formation is that the histones within their DNA have been modified. Histones are proteins that DNA coils around within its double helix structure. Histones have positively charged tails that interact with the negatively charged DNA. These interactions cause DNA to wind around histones. In this illustration, the DNA is shown in purple and the histone is shown in green, with the positively charged tails shown in dark green. Histones can become acetylated as acetyl groups are attached to the tails. If this occurs, the histone tails will no longer be positively charged and therefore no longer able to interact with the negative DNA in the same way. The interaction between the histone tails and the DNA will be much looser, as shown here by the loosely coiled DNA, because the positively charged histone tails are no longer able to bind. This allows for the transcription of DNA to occur much easier because the DNA is more accessible to the various proteins that facilitate transcription. Acetylation of histones might explain why gene F is being highly expressed, because acetylation allows DNA to become more accessible for transcription, and so transcription happens more readily and without as much interference as in deacetylated DNA. An example of a reason that gene F might be expressed at high levels that occurs after pre-mRNA formation is increased RNA stability. In normal mRNA, the poly-A tail is slowly eaten away by RNases, as shown here, the RNase illustrated in red. It starts to be degraded by ribonucleases once the cell recognizes that the poly-A tail at the end of the RNA has become too short, and the mRNA is no longer stable enough to keep around. The ribonuclease is shown here, eating the RNA from the 5' end. It is possible that the mRNA transcribed from gene F has a mutation that causes it to have a very long poly-A tail, or one that is resistant to being chewed away by RNases, as shown here. This means that the cell would keep around the mRNA from gene F much longer and produce much more of its protein because the poly-A tail is not being degraded to the critical point that would notify the cell the time that it is time to degrade the mRNA. So there is a protected poly-A tail, and RNase is not able to access the poly-A tail and begin to break it down. Therefore, the poly-A tail never reaches the critical length that notifies the cell to destroy the RNA, and there is continued protein production of gene F through translation.